Attention student, the remaining parts of this lecture, that is salmonella, we will discuss now. Lab diagnosis of the salmonella. Lab diagnosis of salmonella, the specimen, they are stool, blood, urine and vomitus. If we suspect patient is of salmonellosis, so we will take stool for the culturing or the vomitus for the culturing, and by microscopy under ground staining we will make the smear and we observe gram negative rod, which is confirm that is the salmonella. As we discussed with you that the salmonellosis is the localized disease and it subsided within 24 and 48 hours. Therefore, majority of people in this case do not do any laboratory investigation. The second was major clinical manifestation of salmonella is the enteric fever. This is the most important part of this lecture because in the paper sometime examiner asks you write down the lab diagnosis of enteric fever or the typhoid fever. So listen this part carefully. In the lab diagnosis of the typhoid fever, we use the blood and we perform serological test for the confirmation of typhoid fever. We take the blood and in the blood we try to see the antibodies which is present as well as in some tests the antigen. So these are the serological tests. In the serological test, first is the rapid slide agglutination test. In this test, known antibodies in the form of commercially prepared kit we have, and we confirm unknown antigen from the patient serum. Now known sera and unknown culture are mixed on a slide and observed under microscope a clumping agglutination test. If it shows positive, so we say the test is positive. And the name of this test is rapid slide agglutination test. Once again I repeat, here the known antibody react with the unknown antigen. It means that we have the known antibody in our hand, an unknown antigen that is present in the sera of the patient. So on a slide you put a drop of the sera and apply a drop of antibodies in the form of kit. If clumping occurs, so we say the test is positive. Second test is the Vidal test. In this test, we try to clear the antibodies and we have known antigen prepared kit with us. This is the opposed to the first test. In the first test, we have antibodies but we identified the antigen. In this test, we try to identify the antibodies, not the antibodies, but the titer of the antibodies, which means that amount of the antibodies by a known antigen, which is commercially found in the commercially prepared kits in our hand. Now, there are two types of antigenic prepared kit with us, H antigen and O antigen. One point for your memory, remember this test is positive in second or third week of the salmonella infection. So in the first week, by serologically, we cannot confirm the typhoid fever. Here we take serologically the titer of the antibodies by tube dilution method, as well as we perform this test on a glass slide. In Vidal test, antibody titer against a different salmonella antigen, which is known with us in the form of prepared kit. We take the serum of the patient and serially diluted this serum in a test tube and then apply on a separate test tube for O antigen and H antigen and observe the clumping or agglutination or turbidity in the test tube or on the glass slide, the diluted serum. If we observe clumping in the diluted part of the serum, so we see the titer is high and it is confirmed that the typhoid fever is positive. Now, we said to you that this test is performed after the second or third week. Now, in the third or fourth week, if we again this test, repeat this test, and there is rising titer, we observe, so we say the prognosis of the disease is not good. If the rising titer has become decreased, so we can say prognosis of the disease is good. Now, there is another antigen also present, it's called V1 antigen. If we observe the clumping by this V1 antigen, so the patient is said to be in a carrier state. If you see this slide, so this shows you the method, I mean the procedure, as well as the principle of the typhoid or the Vidal test. Principle of the Vidal test is that antigen and antibody reaction. Remember, unknown antibody. We try to study the titer of the antibody by using known antigen, that is the H and O. Now, the clumping or agglutination between this antigen and antibody reaction shows us the positivity of the typhoid fever. This is the principle of the typhoid fever. Second point for your memory, that remember this test is performed in the second week or the third week of the salmonella infection. If you see the table below, So here serially diluted serum in the table you observe we start 0.08 ml of the serum in a drop of antigen ONH. This is called 1 by 20 ratio. In second diluted at the 0.04. In the third, in the fourth, and at last 0.001 ml of the serum we take and apply upon it a drop of antigen O as well as another drop the other antigen, that is the H, O and H in the separate drop. If we see the table, so 1 by 20 ratio and 1 by 40 ratio, if clumping appear in this part, so we can say the test is negative. And the other diluted material, the diluted serum, do not show the turbidity or the clumping, so we say the test is negative. If the borderline is the 1 by 40, is the 1 by 80 is the borderline. If the turbidity or the clumping observed, we can say 50-50, this is positive, is negative. But the other two dilution, the 3 by 20 or the 1 by 60, this is the confirmative part of this test. If clumping we observed in this part, so we can say the titer is this, and it is the high titer in typhoid fever or the Vidal test, we can say it is positive. Now the third type of test is called typhoid. Typhoid is the modern test where we take a strip, chromogenic strip. When we dip the strip in the serum of the patient, so the change of color we observe on the strip, we can say if the change of the chromogenic substance we observe, so we say the typhoid fever is positive or the enteric fever is positive. This is called typhoid test. This is also a serological test. The fifth one is also the step method, but a plastic cassette commercially prepared available in the lab. This is cassette containing a two type of mark portion, one for IgG antibodies and second for IgM antibodies. If we apply a patient serum upon this strip on this cassette, when the serum touches to this line, we observe the change of the color of this line. If IgG line give discoloration or colored, so we can say the IgG antibodies are present against the salmonella. If the line which for IgM has become colored, so we say that the antibodies is the IgM. Now the importance of these two lines, the IgG antibodies titer if rise in the patient's serum, so we can say it is the acute episode of the enteric fever. If IgM color shows discoloration, so we can say that the episode is chronic and for the longer time the patient is suffering from the enteric fever. Now this is the serological method for the Vidal test. The next, you can write in your paper about this test. This is the PCR, where we identify the antigen as well as the antibodies and the antigen DNA. Antigen DNA we can easily detect it, and we can say the types of the serotypes of the salmonella serotypes, either it is the typhoid or the paratyphoid species, the serotypes. ELISA, another method where we observe the antibodies against salmonella as well as antigen of the salmonella if present within the serum of the patient. So it's all about the Vidal test. Now we again repeat the first part, that is the stool examination. We can perform the test on stool examination in the first week if we suspected that the patient is suffering from salmonella as well as salmonellosis we suspected in the patient. So first is the microscopy, which we discussed with you. Second is the culturing method. So we culture the stool or the vomitus of the patient by different type of culture media. These medias are, number one, first is the McConkey. And the McConkey agar, the McConkey media. If we apply the specimen stick upon the slide, after the 24 hours, we can observe colorless colonies on the McConkey's media. And this is dome shaped, crenated, and centrally black mark present in the culture colony of the salmonella, because the salmonella produce by metabolic activities hydrogen sulfide gas. Second culture medium, which is the selective media, that is the EB. The second culture media, which are the selective media, we can use. It means there is only for the salmonella. It is the deoxychocolate citrate agar, DCDCA, deoxychocolate citrate agar. Second is the salmonella shigella agar. These two are the selective media. Selective mean on this media only salmonella can be grown, not the other species. The third method is the chemical reaction or the biochemical test. For the biochemical test, we used a special type of media which is called triple sugar iron media, TSI media. I hope this media as well as McConkey and this TSI, it was shown by your demonstrator lecturer in the lab to you. In a test tube, this is a yellow color medium, TSI. Must remember, this agar is not used for the growth of microorganism, is only used for the biochemical analysis, that which type of enzyme, which type of metabolic activity is performed by this microorganism. So TSI media is used. If we streak a growing culture on the slope of the TSI media, so after 24 or the 48 hours time, we can see the change of the color of the bud as well as of the slope by this microorganism. So salmonella produce two type of gas. It ferments glucose. By the fermentation of the glucose, production of the carbon dioxide produce crack in the TSI media, colorless track. Second, we observe the blackening in the bottom of the TSI media, which confirm that the microorganism is a salmonella, because this blackening of the test tube of the media that due to the production of the H2S gas. And we have discussed with you the H2S gas is produced by only the salmonella species in the Enterobacteriaceae family, and this due to the use or the biochemical activities that this microorganism use some amino acids, sulfur containing amino acids. So the blackening is occur here. Now this page or this slide we have discussed with you. It shows to you the principle, procedure as well as the explanation about the Vidal test. And again we proceed towards the other parts of this topic. Before to proceed the other part of this topic, once again I repeat you that in the examination paper, this whole procedure they cannot ask you. They ask in the examination paper either bifurcated or in the different parts of the salmonella diagnosis. Sometimes they ask write down the lab diagnosis of the salmonellosis, while in the majority they ask write down the lab diagnosis of the enteric fever or the typhoid fever, or write down the principle of the typhoid fever like this. Now we discuss with you the lab diagnosis once for the salmonellosis. Here we can say the microscopy by stool microscopy as well as culturing of the vomitus and the stool. But in the Vidal test, we only confirm the typhoid fever by the serological test, which we discussed with you. But you can say by microscopy, use the specimen stool microscopy, then culturing, then at last the serological procedure that ended in the PCR and in ELISA.